Thank you, Professor Chen, for that wonderful talk in the field of theranostics and nanomedicine. We have received a lot of questions. Most of them are very basic questions, and some of them are uh, related to your studies, specifically related to your studies. So I'll start with the basic ones first. Um, what is the ideal size of nanoparticles, that is required for any application in terms of nanomedicine or theranostics?
Wow, it's a really fundamental question. Uh, I would say, it's not no short answer to that, but very quickly, and also that's the one reason why we want to launch a new journal, ACS Nano Medicine. To make it medicine, not just a nanoparticle, to have the curiosity to gain new knowledge about the nanoscience, nanoparticles, nanomedicine. It's a medicine. So you have to bear in mind, ask the question whether eventually such a particle or formula can be translatable into the clinic. If money is not a problem, if uh, flow is not a problem. Can we get regulatory approval eventually? Right? Not necessarily everything will be translatable. Not everything will be eventually become drugs. But talking about medicine, you need to think along the way. Um, it's translatable. Also, ask yourself the question: if you yourself or your relatives, your significant others, become sick, are you willing to have your best findings put into yourself or your relatives, right? So those are, it's not joke. It's a very important questions. They have to be, talking about the translation, all the ethics issues, all the translatability. So here, a lot of things we can talk about, but the fundamental one is, are they truly injectable? Okay.
Yeah, thanks for that wonderful answer. Um, Coming to the next question, can we have any in vitro, or are there any in vitro approaches available that will, make sure that there's a better correlation between animal and human studies going forward? Like, you know, let's say
Yes.
short list our final candidates that go into the animal studies, and so that we end up studying only the ones that will show a better correlation going ahead.
Right. Yeah, this question is very important. Recently, NIH and FDA purposely are promoting the in vitro testing, especially the organoids, the 3D cell culture models, very complex, clinically relevant organoid models to minimize animal use, small animals and large animals, especially primate use. Yes, yeah, many such models would be very, very helpful in high throughput screening. Right? That will meet the need, the requirement of the fast advancing AI, machine learning, big data in nanoscience and nanotechnology, right? So a lot of such in vitro models, especially organoids, will definitely become more and more advanced, more closer to human situation before we do animal studies, validating the in vitro findings and eventually clinical translation.
Uh, Coming to the next question, how does the morphology and surface properties of these systems influence the efficacy in chemo or photothermal therapy?
Yes, another very important, fundamentally important questions. Again, no easy answer because people spent decades to study this. Definitely, the shape, the morphology matters a lot. It's a basically it's mechanobiology. You think about Hooke's law, um, the flow, you know, in the tunnel with water, when you have a different shaped material or blocks, right, in the tunnel, you have different shaped nanoparticles in the blood vessel. And there is a stress, there is flow. So the particles will behave very differently. Some of them get stuck on the wall. Some of them extravasate, come out of the blood vessel. Some of them just don't attach to anything or just continue to hang around, right? If you think along this way, you can understand better how the different shapes will behave very differently. Talking about the eventual biodistribution, tumor targeting, and so on. And the same principle not only applies to cancer, but also many other diseases like cardiovascular disease as well. So I probably should stop here for this answer. It's a lot of information, already a lot. For example, a very elegant this group who moved recently to Singapore, Warren Chen, who's executive editor of ACS Nano. He has done a lot of work on different size-shaped nanoparticles, how they behave, and Chet Merkin and many others have spent their life on studying all this.
Thank you. Um, Coming to your study on polymeric mRNA carriers, are there any specific functional groups that are required in those polymers? And also, can natural polymers be used for such applications?
Yeah, again, yeah, I really like this audience. They do raise a lot of interesting and important questions. For a polymer, for mRNA delivery or as an mRNA carrier, definitely will be very different from polymers for siRNA delivery. Oftentimes, I have learned my hard lesson. Initially, we have polymers can have a lot of charges, able to carry the negatively charged mRNA very effectively. Then it comes to the problem, it does not release mRNA readily, right? So unlike siRNA, because mRNA, siRNA, and DNA, they go to different compartments intracellularly. That's why for mRNA, you want relatively loose binding, unlike siRNA. Especially DNA, you need a very tight binding, deliver that all the way to nucleus. In terms of whether natural polymers, they are more clinically relevant, whether it comes from plants or from mammalian cells. Most of those polymers, unfortunately, do not behave well enough as mRNA carriers. People tried, and the performance-wise are suboptimal so far. We might have to modify those natural polymers to some extent to allow effective and safe delivery of mRNA. All right.
Uh, Also, coming to your systems which use polyethylene glycol, have you observed any PEG-associated immune responses or hypersensitivity reactions in your studies?
In animal studies, we do not see that much in the clinic. Of course, a lot of pegylated drugs, especially high molecular weight PEG, not small molecular PEG like PEG 2000. Oftentimes, not that an issue, but like a PEG 30,000, 50,000 in advanced clinical trials, we do see anti-PEG antibody, do see a response to that. And also when the dose of PEG goes high, there might be some cytokine storm as well. But in our studies, the dose is relatively low. We haven't seen that yet.
Thank you. Someone's asking if there is a difference between the nanovesicles that you mentioned, or instead of small unilamellar vesicles, or is it just the nomenclature?
um, Again, interesting question. Right. Even exosomes have gone through name changes, unification, standardization of the nomenclature. Became EVs, extracellular vesicles. EVs and the cell membrane vesicles are different. Put it this way. EVs are secreted. Membrane vesicles are not secreted. They were actually destroyed mechanically or chemically. Then you reform structures. It's outer membrane, not the intracellular exosomes or vesicles being excreted by the cells. So this way, vesicles can be made in large quantity, high yield, but also comes with the risk of those intracellular contents, oftentimes, especially DNA contents and the debris, those garbages coming from this fragmented cell membranes. So it comes to some challenges of purification. Unlike vesicles are really the supernatants. You enrich the supernatants, so you get the EVs. Okay.
Did you observe any difference in the efficacy, or are there any changes that you need to make in terms of design of these nanosystems when you are targeting a benign tumor or a malignant tumor?
I don't fully understand that question. Can you say it again?
I mean, the nanosystems that you have studied, will they have a difference in efficacy when they are used for treating a benign tumor or a malignant tumor? The
Oh,
same systems can be used for both, or do you need to make any changes in design for when it comes to benign or
oh,
malignant tumors?
that that question is convoluted because for vaccine, it's not only the carrier, it's also the mRNA part. And some tumors does not have to be malignant or benign. Some tumors have antigens that are effective. Some tumors, even though malignant, may not have the right antigens, right? So you have to design the mRNA properly, the sequence. Oftentimes, not a single antigen either, right? For example, BioNTech, the German company, has the ongoing phase 3 clinical trial for melanoma. They put 34 different mRNA or antigens into an mRNA sequence. Then they're using lipid nanoparticles to treat. So it's not about whether benign, it's benign or malignant. Then the particles will be more effective or not. It really comes to uh, the immune environment. Okay.
Uh, in the interest of time, I'll take the last question. What do you see as the most critical design parameter that will help nanosystems like carbon dots or something to overcome the endosomal escape hurdle that is there for mRNA delivery?
Um, I'm glad that he brought, this person brought up this particular keyword, carbon nanotube. I've worked on carbon nanotube for many years. Unfortunately, carbon nanotube, because of the size and the shape, it does not come out. So simple answer is carbon nanotube itself does not have hope for clinic. Right? And many other inorganic or semi-inorganic nanoparticles won't have a bright future in the clinic.
Thank you, Professor Chen, for all your answers. It was a wonderful talk. I'll now hand over the session back to Neha.